So a quick run through of what we mentioned last time. We did blood flow through the kidney. We zoomed in to see this functional unit of the kidney, this thing that we called a nephron. We had to sketch the nephron. We will once again label the parts of the nephron and then talked about kind of what each part does. And there was special attention paid to the last part of the nephron because it was lined by cells that were sensitive to hormones. And depending on what hormones were present, these cells could adjust what ended up down here in the urine. And you can kind of see one of the things we did at the end of last lecture was we zoomed in to this part of the nephron, which is what we have over there, and we're going to kind of set that up. It should help us blend a review from last time and then new stuff for today. So there's my to-do list. Any questions about anything last time? The handouts here are just the case study stuff that's new today. I've got the keys to the previous handouts over here if you wanted to glance at any of that stuff. To the nephron. We've got our nephron sketch over here. And this time, let's just start by labeling the parts of the nephron, and then we'll finish the blood vessels that take blood to and from this nephron. We called this part, this kind of Pac-Man looking structure, the first part of the nephron, we called it Bowman's capsule. It also went by the name glomerular capsule because inside of this was the tangled blood vessel that we called the glomerulus. We'll put that in there in just a second. Then there was this part of the nephron that we called the proximal convoluted tubule. That proximal convoluted tubule led us into this long loop of Henle. We'll see that loop of Henle. It's going to have a descending limb where this filtrate is going to make its way down. The loop of Henle, it does a sharp turn and then it's going to make its way up. So we've got our descending limb followed by the ascending limb of the loop of Henle. And then once we follow that ascending limb up, we run into this last set of squiggly lines that we called the distal convoluted tubule. And then that distal convoluted tubule led into this last structure that I'll just label up here as our collecting duct. We said these collecting ducts are going to be shared by multiple nephrons. So I've drawn a nephron over here that's got its distal convoluted tubule hooked up to a collecting duct. But you can also see I've left some space over here. These are representing distal convoluted tubules from other nephrons that are also sharing this collecting duct. They can deposit their filtrate into this collecting duct. And all the stuff that makes its way out of the collecting duct into those minor calyxes is what we consider the urine. So we wanted to see what was in the urine. How do we regulate what's in the urine? So that's what made us kind of look back to see what's going on in the different parts of the nephron. So now we've got it all labeled. We can see that distal convoluted tubule and collecting duct are lined by those cells that are affected by hormones. I've just put a couple of colors there that are going to represent my hormone receptors, and I've even drawn on the other side the names of those receptors so you can kind of see where we're going. We doing okay on that so far, the nephron stuff? Let's label the blood vessels that are going to deliver blood to this nephron. Let's get some blood vessel colors. So far well, it may not be obvious, but this triangular looking thing that these blood vessels are wrapping around, we said was a pyramid. If we look inside the kidney, there are these renal pyramids, and there are going to be blood vessels that kind of wrap around these pyramids. We said the blood vessels that were running right alongside of the pyramids, we called those interlobar arteries and veins. So in this case, my red one is an artery and a blue one is a vein. I'll just label them interlobar artery and vein. So interlobar artery and vein. Then if we follow this interlobar artery up, it's going to kind of make an arc and then follow along the base of this pyramid. The flat part here Those blood vessels we're going to call arcuate arteries and veins. So we've got our arcuate artery and vein, and we can see branching off of this arcuate artery, there is the artery that radiates out into the cortex. Last time we were here, we kind of put a dotted line, kind of a symbolic dotted line right along the base of these pyramids. And those pyramids in the kidney allow us to separate the middle part of the kidney that we called the medulla from the outer part that we called the cortex. I'm sure I'll have to erase those in a second, but it describes the name of these blood vessels that radiate out into the cortex. We called these blood vessels cortical radiate arteries and veins. Cortical radiate artery and vein. Now that we have that in the name, let me get rid of cortex and medulla. We can just recognize cortex and medulla by seeing where those pyramids stop. So everything outside of that is a cortex. This cortical radiate artery, if we follow it out, we're going to see there's a branch off of this cortical radiate artery that's going to deliver blood into this glomerulus. Maybe I can even draw these arrows that show blood flow towards this glomerulus is going to be delivered by the afferent arteriole. The afferent arteriole leads to this glomerulus, that tangled capillary bed, and then away from the glomerulus is a blood vessel that we're going to call the efferent arteriole. I'll draw these arrows that show it's taking blood away from the glomerulus, efferent arteriole. Okay, we're doing good so far. If we follow this efferent arteriole past Bowman's capsule and into this area where it would be found near the proximal convoluted tubule or this distal convoluted tubule, that blood vessel now is going to get the name paratubular capillaries. Paratubular capillaries surround that proximal and distal convoluted tubule, and then we're going to see some branches of these efferent arterioles that don't happen to be found around the proximal and distal convoluted tubule, but instead drop down around this loop of Henle. I'll use a slightly different color. We're going to see that the color changes as we make our way around the base of this loop of Henle, and the other side we're going to draw in blue. It's kind of the venule end. And I didn't draw this last time, you see it in the video, but we're going to see branches of this blood vessel that we should label vasa recta. So these blood vessels that surround the loop of Henle or the collecting duct that are near either of these things we call the vasa recta. Branches of this vasa recta are going to take blood right back into this cortical radiate vein. We could keep the arrows just to show this path that blood takes through the kidneys. We saw this last time in the lecture handout, so we know that blood comes in through the renal artery, it goes through these segmental arteries, and then into the interlobar arteries. Through the arcuate arteries, through the cortical radiate arteries, into these afferent arterioles, and through the glomerulus. It was the afferent arteriole, glomerulus, efferent arteriole, as well as vasa recta and paratubular capillary. These are the blood vessels that interacted with the nephron. So we'll see, we've labeled the parts of the nephron. We need to talk about what each part does. We're going to see that things are going to be filtrated out of the bloodstream, and the nephron is going to reabsorb some things and secrete others. Things that get reabsorbed are going to get picked up by these blood vessels. Anything that gets secreted is going to be secreted by these blood vessels into the nephron tubes. So as another review real quick before we get into the other one, we said Here in Bowman's capsule, this is where you find the glomerulus, and the glomerulus is where we found fenestrated capillaries. Those were the capillaries that had little pores in them. So if water molecules or ions were small enough to fit through those pores, then they were just filtrated out into Bowman's capsule. So anything that was small enough to make its way out came out. Things like sodium ions, things like potassium ions or hydrogen ions. We even include things like glucose molecules that are small enough to fit through there. And we might as well put some water molecules in there as well. So anything that's small enough is going to fit out. Anything that is small enough is going to squeeze out of those holes of the fenestrated capillaries. Not all of that stuff is going to end up in the urine. In fact, we said the urine is mostly, it's going to contain sodium ions and it's going to contain water. And the rest of the stuff that it contains varies depending on what these kidneys are trying to do. So if we go all the way back to the start, we're going to see the first thing after the Bowman's capsule is selective reabsorption that occurs at the proximal convoluted tubule. Let's put some numbers to this. We're going to see maybe the most notable thing is that of this glucose, we're going to see that 100% of the glucose is going to get reabsorbed. That means it's pulled out of these kidney tubules. It's not going to end up in the urine. It's pulled into this interstitial fluid, and then it's going to get picked right back up by those peritubular capillaries. This is how glucose is not wasted. It's reabsorbed into the bloodstream. I think we made a point to say absorption is what happened in the digestive system. It's when we ate the Snickers bar and got the glucose into our bloodstream from, the outside environment. But this glucose molecule, it just came out of the bloodstream. So we're not absorbing it again for the first time. We're reabsorbing it. It just popped out for a second, and then it's going to make its way right back into the bloodstream. In addition to picking up 100% of the glucose, we're going to see that about 99% of the water molecules right here in the proximal convoluted tubule are also going to get reclaimed. So reabsorption of water molecules, about 99%. And the same can go for sodium. Let's see if we can squeeze sodium in there. We'll say about 99% of these sodium ions are also going to end up getting reabsorbed right there in the proximal convoluted tubule. It's trying to reclaim anything that can be used. Of course, the cells can reuse water. Of course, they can use more sodium. And of course, they can use the glucose. Some of these sodium ions, like we said, are not going to end up getting picked up. A few of them are going to make their way into this loop of Henle. We can see that not all the water molecules get picked up. A few of those also make their way into this loop of Henle. And I'll just put some other dots in here that might represent waste, like urea. Put a little bit of that in there. Okay, so we've got through the proximal convoluted tubule. That was our chance to reclaim any of those glucose molecules, mostly, how can I say that again? This was our chance to reclaim, our only chance to reclaim glucose molecules. It was a good chance to reclaim water, and we'll see about 25% of the sodium that gets pulled into Bowman's capsule is going to get reclaimed in this proximal convoluted tubule. As we go through that proximal convoluted tubule and down into this loop of Henle, we're going to see the cell types change on either side of that loop of Henle. The descending limb, maybe as a review question, what was coming out of the cells that line this descending limb of the loop of Henle? What are they really good at extracting out of the nephron tubules? We've got selective reabsorption of things in the descending and ascending limb.
Is the descending water?
Yes, that's right. So the descending limb, it's going to be lined by cells. We don't have to know their name, but what's interesting about them is that they are permeable to water molecules. So water molecules are going to make their way from this descending limb out into the extracellular fluid. And what that's doing is it's leaving behind more concentrated filtrate, all this stuff in the nephron tubes. As that water is left behind, we've got a higher concentration of these sodium ions. So now they're in real high concentration because we've just removed a lot of that water. We'll still put the waste in there. And even though we've pulled out a lot of water, we can see there's still going to be some water that ends up in the urine. So I'll put some water molecules back in there. There's just not nearly as diluted as the filtrate was before we go down this descending limb. So those water molecules that get pulled out down the descending limb are reabsorbed by this vasa recta. That's going to help maintain blood volume, and that regulates blood pressure. So we make this turn around the loop of Henle. Now we've got this really concentrated filtrate. It's got a bunch of high concentration of sodium ions in it. And as we go up the ascending limb, the cell type changes. So no more water is going to be able to seep out of the nephron tubes. Instead, what's going to be pulled out of the nephron tubes? I've got the green color, so that's a clue. Sodium, yeah. And I've kind of barely drawn the difference here. There's a thin and a thick segment. Both thin and thick segments are going to be pulling out sodium ions. It's just that the thin segment does so passively. And that's because these sodium ions are still in very high concentration down low at the nephron tubule, so that they easily go from high concentration into an area of low concentration. It's like falling down the stairs. It doesn't take as much energy as it does to walk up the stairs if you're going against that concentration gradient. That's what's happening in the thick segment. The sodium ions are still getting pulled out, but they're in relatively low concentrations. So to pull them from an area of low concentration towards an area of high concentration is going to involve some ATP. I'm not going to put that anywhere on here because I'm not going to ask that. I just, maybe as a reflex, I have to say it. Sodium reabsorption, passively at first and then actively at the end. So by the time we get up into this distal convoluted tubule, there's still some sodium ions in there, but they're not nearly as highly concentrated as they were before we went up that ascending limb. We said there's still some water molecules that are in there, because if those water molecules were not absorbed on that descending limb, there's no way for those water molecules to come out along that ascending limb. So here we are in the distal convoluted tubule and the collecting duct. We've got relatively diluted urine. There's still some water. There's still some sodium ions in there. And we can regulate what ends up in the urine because the cells that line the distal convoluted tubule are going to be sensitive to certain hormones. I've just drawn them here in a different color, but this is maybe where we can make our way to the other side of the table, or sorry, other side of the board. And maybe you can help me remember the names of these cells. There are some of these cells that line the distal convoluted tubule and collecting duct, and I've drawn them in kind of a lighter color orange. They have receptors on them for hormones like antidiuretic hormone and aldosterone. And we kind of see what's going on if we look inside that. So we've just zoomed in here to our collecting duct. We can see those three types of cells that are hooked up to the collecting duct or distal convoluted tubule. So their apical surface faces the collecting duct, and then their basal surface, the opposite side of the cell, faces that vasa recta. If we were in the distal convoluted tubule, this would be called a paratubular capillary. But we're in the collecting duct, so we're calling it the vasa recta. There is, well, let's just name these cells first before I go any further with this. What did we call this cell that has receptors for antidiuretic hormone and aldosterone? Let me get my color. It was a principal cell. And these principal cells, we see where they're found, distal convoluted tubule and collecting duct, and their job is to regulate things like water and sodium ions. And there's two different receptors that do this. We looked a little more closely at the details last time. We said if there was a situation where we wanted to boost blood volume, one of the ways to or boost blood pressure would be to pull more water into the blood vessels. And one way that we could get more water into the blood vessels is by releasing this antidiuretic hormone. This antidiuretic hormone is going to basically prevent water from ending up down here in the urine. It's going to reclaim that water and keep it in the blood vessels. So antidiuretic hormone, it's coming through this bloodstream. It hops out of the bloodstream and onto this receptor. Principal cells have these receptors for antidiuretic hormone, and once an antidiuretic hormone docks, to give you the Cliff Notes version of it, there's a cascade of protein events that lead to an activation of a water channel. It's called an aquaporin, and we mentioned last time there are several different aquaporins. Aquaporin 3 and 4 are always open, and they're on the basal side of the cell that faces the blood vessel, but not the collecting duct side. This is where the water is, so the other aquaporin protein that's locked inside the cytoplasm of a cell can't get to the water. It's inactive at this point. But antidiuretic hormone, if it's present on the receptor, is going to lead to the activation of this second type of aquaporin protein. If these are activated and inserted on this tubal side, the collecting duct side, that now gives a route for water molecules to be picked up by these principal cells. And there's already a channel for water on the basal side of the cell, so water can just make its way from the cytoplasm of the cell into this interstitial fluid. Once again, I just wanted to write these terms. The fluid inside the cell we call cytoplasm. That fluid outside of the cell we called a couple of names. We called it extracellular fluid, which is true, it's outside the cell. And we also referred to it as interstitial fluid, the fluid in between cells. So we've got both terms here. I'm going to try and distinguish plasma from interstitial fluid. Interstitial fluid is the fluid outside of cells, but it's not the fluid that's inside blood vessels. That fluid we called plasma. In fact, let's maybe put this note up there that may clarify it. We could say that plasma and interstitial fluid, both of these things are examples of extracellular fluid. This ECF is kind of a broad category. It refers to all of the fluid outside of the cell. Some of that fluid is inside blood vessels, and we would call that type of extracellular fluid plasma. The other types of extracellular fluid that you see just kind of in between the cells, we call that interstitial fluid. So that's a distinction between the types of extracellular fluid. Just trying to clarify that, because I just mentioned these water molecules went from the collecting duct into the cytoplasm, and then they are pulled out of the cytoplasm of the cell into this interstitial fluid, and then they're picked up once again by the blood vessels. Anyway, so this is one way that antidiuretic hormone reduces the amount of water that ends up in the urine and increases the amount of water that stays in our blood vessels. So aquaporin 2 gets activated, aquaporin 3 and 4 were always there. Let me get that out of the way real quick. Now we'll mention the other thing that principal cell does. It has receptors for aldosterone, so we can imagine aldosterone being sent this chemical signal through the bloodstream. If aldosterone docks to one of these receptors, it's going to activate this aldosterone channel that I just drew with the big letter A. If we activate this aldosterone channel, the thing that we needed to remember about this aldosterone channel, it's like a revolving door. It prevents sodium from being excreted in the urine. It's going to reclaim a sodium molecule. So here's this sodium that's about to end up going down in the urine. It now has a route, now that we've activated this thing. It's stuck on the plasma membrane. This sodium can have a way in, so it doesn't end up in the urine, and the sodium is going to make its way out of the basal side of this cell and back into the bloodstream. So aldosterone prevents sodium from ending up in the urine. It increases the amount of sodium reabsorption into the bloodstream. There was that part, but then we added last time that aldosterone, it's like a revolving door. So anytime a cell is going to pick up a positive charge, it's going to have to balance that out. It's going to trade it off by getting rid of a positive charge. For every sodium that gets reabsorbed, we're going to see there's a potassium ion that gets secreted. So sometimes there's potassium ions that end up out here in the urine. One way could be if you have prolonged working of aldosterone. It holds on to sodium, but it gets rid of those potassium ions. Okay, so principal cells are the ones that have receptors for aldosterone and antidiuretic hormone, and those are the cells that really regulate sodium and water, but there were these other two types of cells that had something to do with regulating the pH of the bloodstream. I don't have them labeled here, so I guess you could just tell me. We'll label them, and then based off of the label, we'll talk about what those different cells do. I got them in a slightly different color. What are some names of these two cells? Yep, we could have listed them in any order. So we'll put the first one as a type A, what type of cell? Yeah, type A intercalated cell. Type A intercalated cell. The other one is a type B intercalated cell. So type A and type B intercalated cells. If we look at what's going on inside either one of those, we said maybe I could ask which one of these is going to be activated if the blood happens to be too acidic. So your blood is too acidic. One of those cells can help out. Which one would kick into gear if your blood is too acidic? Coffee break. There's a clue right in the name. That's the clue. They were really named based off of the fact that in humans, blood tends to be more often, the problem is that the blood tends to be more acidic than basic. So type A intercalated cells are going to be activated more frequently than type B intercalated cells, but the way that it's easy to remember, type A, you can think of the A tells us that these cells respond when the blood is too acidic. Type B tells us that these are the cells that respond, the type B cells respond when the blood is too basic or too alkaline. Let's visualize that real quick. Here's our bloodstream. And in the picture of our bloodstream, if we try and draw some acidic blood, this is blood that just has too many of these free hydrogen ions in it. These ions you find in the blood are this bicarbonate ion that's negatively charged and these positively charged hydrogen ions. Right now there's more hydrogen ions than we have hydroxide ions, so we've got some blood that's acidic. This is problematic, but not to fear. Type A intercalated cells are on the scene. And if we see what's going on there, if I can find my marker, there it is. And I'll get rid of that cytoplasm word. We said that inside the cells, there is water inside the cells. There's also carbon dioxide inside cells, and that water mixes with carbon dioxide to form this unstable molecule that we call carbonic acid. This, any acid is going to break apart and donate a hydrogen ion into solution. So this carbonic acid is unstable and it's about to break apart. We're going to see that, like its name suggests, one of these things that it's going to break apart into is a hydrogen ion. We've already got too many of those in the bloodstream, so we don't want that. But the other half of this molecule is bicarbonate. And as this thing disassociates, the type A intercalated cells know enough to cause this hydrogen ion. We don't want that in the bloodstream. Let's get rid of this hydrogen ion. There's already plenty of those. So that ends up down here in the urine. And the other half of this bicarbonate, ends up in the interstitial fluid, a type of extracellular fluid for a second, and then it's picked up by the bloodstream. So just a couple of these things getting picked up. We've now neutralized the pH of that blood, so it's no longer in its problematic state. The pH of the blood has to stay on a very narrow range of pH. So it's very important that these chemical buffers can respond immediately if the blood gets too far out of whack. So the first thing on the scene, and this is the case study that we're going to do, the first thing on the scene that can respond to the blood's pH getting too far out of whack are going to be these chemical buffers. And the main chemical buffer is this bicarbonate. So when it disassociates, we're going to see that some cells can put more base into the blood, and they get rid of the other half, which is the hydrogen ion. We said this last time. We said that you can think of the fact that if the blood is too acidic, the urine is also going to be acidic, because the kidneys are trying to compensate for the fact the blood is too acidic. We've got too many hydrogen ions here, so we're going to start getting rid of those hydrogen ions in the urine. That's great. There's only one other thing that we said to that, and it was just what I mentioned up here. Anytime a cell is going to get rid of a positively charged ion, there's a trade-off. It has to balance that by, if in this case it was picking up a positively charged ion, so it had to get rid of one. What we've drawn here is this cell has just gotten rid of a positively charged ion, so it has to pick one back up. There's potassium ions that are in the urine. I guess I should say consistent with my potassium color, but I guess I'm not going to be able to. Let's see, as this hydrogen ion is excreted from the cell, what the cell picks up is a positively charged potassium ion. I'll just put it in a different color than the hydrogen so we can distinguish it. That's how the cell kind of maintains its overall charge. It's gotten rid of a positive charge. It's picked up this potassium, but this potassium is going to travel through the cell and be picked up by the bloodstream. So there's just a side note with these intercalated cells. They're adjusting the pH of the bloodstream, but there's also going to be this side effect of either reabsorbing potassium, or in the case of type B cells, we're going to see these are going to secrete potassium. Just remember, potassium is always linked with the hydrogen. These are two positively charged ions, and the cells have to swap one positively charged ion for another positively charged ion. Let's see this same thing again, except flipped in reverse on the type B intercalated cells. These things operate the opposite way of the type A intercalated cells, and they respond to situations when the blood is too alkaline. Are we doing okay with this one so far? I guess I should ask. Yeah?
It doesn't matter which positively charged ion it is?
It does. It has to do with the special channel that's getting rid of the hydrogen ion. And the special channel, the way that these intercalated cells do it is that they pick up a potassium for every hydrogen ion. If maybe that's a great question, if there was some type of mutation in a person that caused their intercalated channels to swap calcium for a potassium, like what would that do in terms of the physiology of that person? That's interesting to think about. So like what in our evolutionary history, locked us into just being able to swap that particular ion? I don't know if I could go any further than that, but that's a good thought. Both of these cells do the same thing. They're going to always switch a potassium for that hydrogen ion. And so let's look at that down here. In the type B intercalated cells, these are again going to respond if it's a situation like this, where we've got too much of this bicarbonate in there and not enough of those free hydrogen ions. This is also problematic. This is alkaline blood. Type B intercalated cells are here to help. So this time when the carbonic acid disassociates, we're going to see that the hydrogen ion is going to be needed. So we wouldn't want to secrete that into the urine. This time it's going to be pulled into the bloodstream. So this is reabsorption of these hydrogen ions. That neutralizes the blood, but like we just said, in order to get rid of one of these positively charged ions, the cell's going to have to pick up a positively charged ion. Once again, it's a potassium ion, and it gets it from the bloodstream. So these type B intercalated cells, they're going to pull potassium out of the bloodstream, and potassium is going to end up in the urine. The other side of that, like we said, the other thing that ends up in the urine of the type B intercalated cell is the other half of this carbonic acid, that bicarbonate molecule. There's already too much of that in the bloodstream, so we don't want this going into the capillary. This is instead going to get secreted into the urine. And it follows that if the blood is too alkaline, then the urine is also going to be alkaline because these cells are getting rid of that bicarbonate. The other side of that story, like we said, is the positive ion that gets swapped for hydrogen. Okay, I'm going to leave that up there. Sip on some coffee, and I think we're ready to proceed into some slides unless there's questions that I've stirred up. Yeah.
One question.
Sure.
So type A pulls potassium from the urine,
Correct.
and type B pulls potassium from the blood vessels.
That's right. So long periods of type A cells working could lead to a situation of hyperkalemia, where you've got too much of that potassium in the bloodstream. Type B cells working for too long could end up stimulating hypokalemia because you've reduced potassium ions in the bloodstream. That's right. Neurons need them both. I think we've done, well, I don't want to brag on everybody here, but I think we've done pretty good with our review. So that might allow us to click through these slides with some ease. And I'm going to get out of the way and take a seat over here. I think there's only like 20 slides. I'll try and point to the stuff that's on the board as we go through, but stop me at any point if there's questions. The first couple of slides that we look at here really just talk about water and where we find it in the body. This is our, to go back to the title, fluid and electrolyte balance. And that ties into what we're thinking about, because if we look at what ends up in the urine down here, it's all about how do we regulate the fluid, the water that either ends up in the urine or gets pulled back into the bloodstream, and how do we regulate the ions like sodium or potassium. We'll start with the water. This mentions that if we take 100% of the water in your body, on average, that's going to end up being about 42 liters of water. And on average, it accounts for about 60% of your body weight. That might not be exactly 60%. It could be anywhere from 40 to 80% of your total body weight. And what adjusts that range is the percentage, most often the percentage of adipose tissue. Just wanted to point out that most of the tissues of the body have a relatively high water content. Plasma, we said, was almost entirely water. Skin and muscles and internal organs are about 80% water. The skeleton even, so compact and spongy bone make up those are about 22% water. The cell that is made up of the least amount of water is adipose tissue. So lean individuals that have less adipose tissue are going to have a higher body water percentage. They would be up there closer to the 80% body water. And then the other note is that as age increases, body water decreases. Which sounds like a bummer, but remember, old age is a gift denied to many. So we should all be so lucky to deal with that, hopefully. All right, so moving on. This is what I drew on the board, and it just tries to make a distinction between this extracellular fluid and the intracellular fluid. Intracellular fluid is easy. We call that cytoplasm. So the fluid inside the cell, cytoplasm or intracellular fluid, is going to be where most of the body water is located. If we go back to that 42 liters of water, most of that, about 70% of it, is going to be found inside cells in the cytoplasm. So about 30% of it is going to be that fluid outside of cells, and I'm coming back up to the board because that fluid outside of the cells could either just be hanging out in between the cells, in which case we could give it the more specific term of interstitial fluid, or maybe it's been picked up by a capillary, in which case we would call that fluid the plasma. What would we call this fluid if it was picked up by a lymphatic capillary? Lymph, yeah. So it just gets different names depending on where we find that water. So if it's inside the cell, it's cytoplasm or intracellular fluid. If it's outside the cell, it's in one of these compartments that we consider extracellular fluid, either the plasma or that interstitial fluid. And just like the picture is trying to show you here, there's more. If we look at the extracellular fluid just by itself, most of that extracellular fluid is outside of the cell. It's this interstitial fluid. It's not within blood vessels. So plasma only constitutes about 20% of the extracellular fluid. I don't want to get caught up in the percentages of this. If any percentages, just know that about 70% of the body water is locked away inside cells, and the other 30% of that water is outside of the cells, either in interstitial fluid or in plasma. Now, the two types of, like I said, just the extracellular fluid that we mentioned, there is the stuff in the plasma and this interstitial fluid. What this is trying to show us, and I guess I just have to jump in front of these slides, there are no special channels or gates between this interstitial fluid and the plasma. So ions that I've been drawing back and forth here just passively go between this interstitial fluid and the plasma. There's no special protein channels like the aquaporins or any of those things that you see on cell membranes. Cell membranes have all kind of special protein channels that really can regulate the stuff that you find, the ions and proteins that you find inside the cell versus outside the cell, which is how we end up with this next slide that shows us these two columns are, one column is showing us the relative ions that we find in the plasma. And you could see how similar the concentration of ions in the plasma matches up to the concentration of ions in the interstitial fluid. It's mostly sodium, a lot of chloride, a little bit of this bicarbonate. Maybe one of the main differences between interstitial fluid and the plasma is that inside the plasma, and we drew this last time when we were drawing a cross-section through the capillary, we listed things like formed elements, there was the plasma, there were ions, and then there were plasma proteins that we found obviously in the plasma. Those were too big to fit through the little spaces in between the capillary walls. So plasma proteins should not have leaked out into that interstitial fluid. The ions were small enough to leak through. So we just have passive transport. That's why things are very similar between the plasma and the interstitial fluid. But when we get inside the cells, you can see the ion concentration is completely different. There's a very high concentration of things like potassium ions, very little sodium. This takes us back to talking about neurons last time, which we don't really have to know the differences between all the ions. I just want to show out how there's very similar ion concentrations between interstitial fluid and plasma, drastically different ion concentrations once we get inside the cell. The main ion concentration that we focus on going forward are those bare hydrogen ions. Because the blood, we said, has to be kept in a very narrow range of those free hydrogen ions. So there are all kind of buffers that we are going to see that help regulate or prevent there from being too many of these free hydrogen ions in the bloodstream. This takes us to a couple of slides that we have here that once again try and mention what an acid is and what a base does. This example of an acid that we've drawn on the board is a good example because it donates, it breaks apart and gets rid of one of those hydrogen ions. And in the picture that we can see, it looks like there is hydrochloric acid that has been dropped into this top beaker, and it breaks apart into a bare hydrogen ion and then a chloride. So any acid is going to donate these free hydrogen ions. And an acidic substance is one that has an excess amount of these free hydrogen ions. Down here in the bottom beaker, we can see that a buffer has been added to this acidic solution. Just like we've drawn on here, this carbonic acid, it breaks apart into part acid and part base. And those can either, this could combine with a negatively charged ion and be removed. That could end up combining with a positively charged ion. And we're going to see both of them are used just to try and wrangle up any of these free hydrogen ions. This just shows us that the narrow range of blood pH that is tolerable is anywhere from 6.8 to about 8. Anything above that it becomes fatal very quickly. So a normal range, something that is easily tolerated, is anywhere from 3.5 to 4.5. Outside of that, so anything above that gets us into this alkalosis range. Above 8 is likely fatal. Anything below the normal range is in acidosis. Anything down to 6.8 is, or below that, is likely to turn fatal. The body has ways of compensating if the blood becomes too alkaline or if it becomes too acidic. And we talked about some of those cells that can respond when the blood is too acidic or too alkaline. And there's some other compensatory mechanisms that we'll mention in this PowerPoint. So I just gave that away. Normal ranges are, normal pH ranges of the blood are from 7.35 to 7.45. And we said below those normal ranges is acidosis, above those normal ranges is alkalosis. You're going to see 4 individuals in the case study today, and the first thing that you're going to look at is what is the pH range of their blood. If it is above 7, try this again, if it's above 7.45, you can immediately label those people as being in the situation where they are in alkalosis. There's a couple of reasons maybe why their blood could be too alkaline. Maybe it's the respiratory system that's failing. We talked about, well, we'll see some situations in the respiratory system that can lead to the blood becoming too alkaline. I think I talked about when there was a person that was hyperventilating because they were about to take an exam, and it was, I guess maybe a very stressful exam, and so they're stressing out and they're hyperventilating, they're blowing off all of their CO2, and that blowing off of the CO2 led to their blood becoming too alkaline. And so that's just an example of sometimes the lungs can cause the blood to become too alkaline. We'll see there are metabolic reasons why your blood could be too alkaline. Maybe one more common reason would be if somebody has prolonged vomiting. If you've ever been sick and vomited for a long time, at first you might vomit up whatever you ate most recently, which is why I can't eat shrimp anymore for a while. I love shrimp too, but anyway, if you keep vomiting for a prolonged period of time, you start to get to that stomach bile. And if you keep vomiting up this stomach bile, you're basically vomiting up your acids. And so what is left behind is more alkaline blood, because you basically have gotten rid of your free hydrogen ions. So prolonged vomiting is one way that your blood can be too alkaline for metabolic reasons. Maybe another reason, which is not common, but if you were to do like eat too many Tums or eat too many Rolaids, that's another reason why your blood could be too alkaline for metabolic reasons. Or I don't know, some of those like murder mystery shows where somebody, like the wife sneaks some Drano into the husband's drink or something like that. Sometimes somebody just has enough and tries to end their life by drinking Drano or something like that. That's a terrible scenario. But if you were to do something like that, ingestion of a lot of excess base like Rolaids or Drano, something like that, that can make the blood too alkaline. Those scenarios will be helpful when we get to the case study, just not random terrible thoughts. But there's also ways that the blood can be too acidic. Like we said, if you hold on to your breath and you refuse to exhale, then that carbon dioxide builds up in your bloodstream. And as we get more carbon dioxide, we're going to get more of these free hydrogen ions. So the blood can be too acidic for respiratory reasons because you hold on to that CO2. Or the blood could be too acidic for metabolic reasons, like the case of my dad that's a diabetic. His cells can't use glucose as a fuel source. They don't have the way to get glucose into the cells. And so maybe the best way that being diabetic was described to me, it's a case where your cells are starving in the presence of plenty. They're totally surrounded by glucose, but they don't know it's there because it doesn't have a way to get into the cells. So the cells are screaming, I'm hungry, I need some fuel, I need some glucose. And you keep ingesting glucose, but the cells are still screaming that they're hungry because they can't get the glucose into the cell. So what happens is the cell starts breaking down other things like proteins and lipids as a fuel source. And if you start breaking down proteins and lipids as a fuel source, it can start to increase the hydrogen ions in the bloodstream. So those are cases of metabolic acidosis. There's metabolic alkalosis, like eating too much Tums or drinking Drano. And then there's lung reasons why, like we said, you could either hold on to your breath and refuse to exhale. That would lead to acidosis. Or you could hyperventilate and blow off too much CO2, and that could lead to alkaline blood. Coffee deficit. All right. Now, being in a situation where your blood is too acidic is going to cause the nervous system to slow down. My dad sometimes can't make decisions, he can't, his brain is just slowed down if his blood is too acidic. Versus the other situation, if you are in alkalosis, it tends to make the nerves very sensitive and jittery, overactive nervous system. Obviously, the pH of the blood is going to affect enzyme activity. But the other thing, like we just have seen here on the board, let me get out of the way, is that if the blood is too acidic, this situation, it's also going to lead to this indirect effect of potassium levels being adjusted. Those duct cells that we just talked about, if we're in a situation where there is excess hydrogen secretion. So hydrogen secretion is going to lead to potassium reabsorption. And this is what we're doing over here in type A cells. We've got hydrogen secretion. We're getting rid of hydrogen in the urine, so that's going to lead to potassium reabsorption. Type A intercalated cells or acidic blood can lead to hyperkalemia. And alkaline blood, the second part down here, so if our blood is too alkaline and we're in a situation where we need to reabsorb this hydrogen ion, which is what we're doing in these type B cells, as we reabsorb these hydrogen ions, it causes us to secrete potassium. So now we're getting rid of this potassium ion. Okay, so just mention it, when the blood is too acidic or too basic, there are cells that can regulate for that, but there's a side effect of also either holding on to or getting rid of too much of this potassium ion. And like I've drawn for us into the cells, this is just reminding us where most of this bicarbonate comes from. The bicarbonate and these free hydrogen ions really come from the carbon dioxide combining with this water molecule, forming this unstable carbonic acid that we can see immediately disassociates into this free hydrogen ion or this bicarbonate. So how we drew it here on the board is sufficient. Here's where we get into the case study. This is the first one that we don't have to actually do it yet. I'm still setting it up. In fact, I'm going to show how we work our way through these problems. And I'll do the first one together, and then maybe we can break up into groups and work on the other three. But as we are about to approach this case study, what we want to think of is that the body has ways to respond if it's in a situation where the blood is too acidic or if the blood is too basic. And the ways in which the body can respond, it's always a three-tier response. The first phase of that response happens immediately, and it's done so by these chemical buffers. They're present, they're on the scene, so if we start to be in a situation where our blood gets too out of whack in terms of pH, the first thing on the scene are what we call these chemical buffers. So the notable thing on the screen here is that the first mechanism to respond and help regulate or change the pH of the bloodstream are these chemical buffers. The main one that we have talked about is this bicarbonate and carbonic acid buffer. Well, let's look at the beakers here to see what's going on. The top two beakers are showing us a solution that has no buffers in it whatsoever. So it looks like we just have some salt water, and we dropped 3 drops of this hydrochloric acid into salt water. In an unbuffered solution, we've gone from a neutral solution having no free hydrogen ions to now having 3 of these free hydrogen ions. So we have increased the acidity of this solution just by putting 3 drops of hydrochloric acid. And if we had a buffered solution like we do down here, one of those molecules floating around in the solution is this carbonic acid and bicarbonate buffer. So there's no free hydrogen ions in this solution initially, and as we drop 3 of these hydrochloric acids in there, this hydrochloric acid is going to disassociate into a hydrogen ion and a chloride, but with these buffers, the base end of the buffer can bind with those free hydrogen ions and lock them up into this carbonic acid again. So it can go back and forth between bicarbonate and carbonic acid, either donating a hydrogen ion or picking up a hydrogen ion. In a buffered solution, you can see how it has reduced how drastic the shift in pH was. In an unbuffered solution, we went from a pH of neutral all the way to 3, and now we've just changed it by a factor of 1 hydrogen ion. It's reduced the shift in pH. Here are some common buffered solutions. The main one that we talk about, it's the most common one that is responsible for fixing blood pH problems. It's the chemical buffer that you find in the blood plasma, this bicarbonate carbonic acid buffer. There are other ones in the body, just to mention them. There are some parts of proteins that are able to pick up free hydrogen ions or donate hydrogen ions. On that hemoglobin molecule, there are some parts of the hemoglobin that pick up hydrogen ions. I'll just go back to this slide for a second. There's a free hydrogen ion. there's a part of the hemoglobin molecule that's picked it up. Not that we have to really focus on that. Some parts of the hemoglobin are acting as buffers to wrangle these free hydrogen ions. And even in the kidneys, there are chemical buffers that work based off of a phosphate. The main one that we focus on here is the plasma chemical buffer. The next slide shows us that chemical buffer of the plasma, carbonic acid and bicarbonate, the main one that we talk about. Carbonic acid is the acid part that has a hydrogen to donate, and the bicarbonate is the base part. It's looking to pick up one of those hydrogen ions. Just for reference, I gave you those other 3 chemical buffers, and you can see the acid part that has a hydrogen ion to donate, and then the base part that picks up a hydrogen ion. But this one is the first one to show up on the scene to adjust any shifts in the pH. The bad news is that it is not, you don't have an infinite amount of this bicarbonate carbonic acid buffer. There's a limited amount. And if the shift in pH is too drastic where these chemical buffers can't immediately fix it, well then the first line of defense is insufficient at fixing the change. Good news, you've got 2 more lines of defense that can step up and help adjust the pH of the bloodstream. Our second line of defense, if the shift isn't handled immediately, falls on the lungs. The respiratory system can also compensate for the pH of the blood. We mentioned this when we got to the respiratory system. It simply has to do with holding on to CO2, hypoventilation, or getting rid of excess CO2. We call that hyperventilation. The lungs are going to kick in after a few minutes of the chemical buffers not working, but the lungs are going to have less than an hour to compensate. I've mentioned I've seen this before when my dad has broken into this uncontrollable hyperventilating things, and that's his body's mechanism of trying to compensate for the fact that his blood is too acidic. His blood is too acidic because he's a diabetic, and his lungs are working, so his lungs can actually try and compensate for the fact that his blood is too acidic. Here's the scenario. If the blood is too acidic, what the lungs do is they increase the respiratory rate and they increase the depth of breath. So it's not shallow breathing, but it's deep, rapid breathing. Like that. You're really trying to blow off that CO2. And like we said, the more of the CO2 that you get rid of, then the less of these hydrogen ions you're going to have. So the second bullet point here points out what the lungs would do if the blood is too alkaline. If there's not enough acid in the bloodstream, then the lungs are going to respond by decreasing the depth of breath and decreasing respiratory rate. If the blood is too alkaline, we want to increase carbon dioxide levels because that's going to give us more of these hydrogen ions. So you'll see somebody that is in metabolic alkalosis. Their blood is too alkaline for metabolic reasons. Their lungs are fine, so their lungs can help try and compensate by holding on to that CO2. Okay, great. So now we talked about how the lungs are the second line of defense, and they have about 30 to 45 minutes to correct the pH of the blood. So they work on minutes to hours. And if the respiratory system isn't able to compensate for the pH of the blood, we get to our third and final phase of this response, and that has to do with the renal system, the kidneys, these cells right here that can help regulate if the blood is too acidic or if the blood is too alkaline. And I should just say this, one quick thing. If you are in a situation where you have like some type of chronic obstructive pulmonary disease like emphysema, or if you've got some type of lung disease, it could be any kind of lung disease where your lungs are not working properly, then you don't have this second line of defense. You've got chemical buffers, lungs are out of commission, you go straight to the renal system. This becomes your second and final line of defense if you have any type of lung disease. So anyway, let's assume the lungs are fine. So you do have lungs that can either increase or decrease respiratory rate and adjust pH of the blood. But if that's still insufficient, then we move to this renal system, the third line of defense, the type A and the type B intercalated cells. Since we have them on the board, we can see we can try and make sense of what we have up here on the screen. The kidneys are going to have hours to days to compensate. So these can, it takes them, this isn't immediate to work. It doesn't even work in 30 minutes. It might take hours to a couple of days for these type A or type B intercalated cells to really affect the pH of the blood. Here's what happens. If the blood is too acidic, and this is what we're seeing right here. Just think type A intercalated cells. If the blood is too acidic, we're going to see secretion of the hydrogen ions. Secretion means that we're getting rid of it in the urine. There it is. It's getting secreted into the kidney tubules and moved out of the body in the urine. And what we're going to end up reabsorbing are things like the bicarbonate to neutralize all of those hydrogen ions. And then the side effect, the other thing that we end up picking up, is the potassium ions. I don't think potassium ions are mentioned here. But we know that if the blood is too acidic, if we're getting rid of those hydrogen ions, we're conserving bicarbonate. These go the opposite way. So we can see hydrogen ions one way, bicarbonate the other way. Maybe what we can add to this slide, since we already know, potassium ions are going to follow. Potassium ions are going to go the opposite way of wherever the hydrogen ion goes. Different directions. Same thing down here, going opposite ways. So the bottom bullet point, if the blood is too alkaline, this is describing the type B intercalated cells. If we have blood that's too alkaline, then we're going to decrease how much hydrogen we secrete. We're not getting rid of any of that hydrogen. Instead, that hydrogen is being reabsorbed. And instead, we're going to excrete that bicarbonate. The blood already has too much bicarbonate in it, so whenever this carbonic acid splits, we get rid of the bicarbonate. We hold on to that hydrogen ion. Okay, so those are the 3 lines of defense. We're about to see a case study. There are 4 individuals in the case study, and I'll go ahead and say that there is one individual that is going to suffer from each of these 4 situations. So let's describe these situations real quick, because I forget the names of the 4 individuals in the case, or I forget who has what in the case study. One of these individuals in the case study is going to have respiratory acidosis. It's a situation where the blood is too acidic due to respiratory reasons. And I already said one way that the blood can be too acidic due to respiratory reasons is because you've increased the CO2 in your bloodstream. You do that by holding on to your breath, decreasing how many times you exhale. Hypoventilation is what I'm trying to say. Holding on to that CO2 causes the blood to become too acidic. The opposite is true. The respiratory system can also cause the blood to become too alkaline simply by blowing off too much CO2. So hyperventilation causes that excess loss of CO2, and that causes the blood to become too alkaline. Third scenario, I mentioned it, this metabolic acidosis. This is what my dad suffers from because he's diabetic, and that's a situation where your body now, for metabolic reasons, is too acidic because in the case of diabetic, you're breaking down lipids as a fuel source, and so the breakdown of lipids just produces a bunch of these free hydrogen ions. So the blood becomes too acidic because lipids are your fuel source. Severe diarrhea is one thing that can happen. I mentioned, I don't know that I mentioned this yet, but we do have a case study today where there's somebody that has severe diarrhea. They went on vacation. They drank water they weren't supposed to drink. They got prolonged diarrhea, and then they were dehydrated. And they also had an acid-base imbalance due to this prolonged diarrhea. When we were talking about the digestive system last time, this is maybe a good review. We said when we get into the large intestines was trying to reclaim water, and it was trying to reclaim things that could be reused. And one of the things that was reclaimed in the large intestines were these bases. We said that the small intestines doesn't like intense stomach acid. So there is the pancreas that can do 2 things. It can either release bases that neutralize stomach acid if you skip a meal, but the pancreas also can produce any of those digestive enzymes if you eat a big meal. So the pancreas has to constantly be regulating the pH in the small intestines. And that changes depending on if you're eating a meal or if you go long periods of time without eating a meal, then that stomach acid drips into the small intestines. But the pancreas can neutralize that stomach acid by putting a lot of base, these chemical bases, into the small intestines. Those chemical bases are reabsorbed in the large intestines before they're excreted out. So the job of the large intestines is to reclaim some of these chemical buffers, particularly the base part. Now, having said that, if you are having this prolonged diarrhea, then that you're having diarrhea, it's loose, watery stool because your large intestines is not reclaiming that water, and it's also not reclaiming those bases that the pancreas has been releasing to neutralize that stomach acid. So what you're leaving behind is blood that is much more acidic because you have not reclaimed those bases. You haven't reabsorbed those pancreatic bases. So diarrhea, being diabetic, renal failure, or exercise, these are all things that can lead to metabolic acidosis. Then metabolic alkalosis is more rare than metabolic acidosis. That's much more common than this metabolic alkalosis, but it's not impossible. Like we said, prolonged vomiting can lead to this metabolic alkalosis. Your blood is too alkaline because you vomited up those acids. In diarrhea, you're losing bases. In prolonged vomiting, you're losing acids. So you're leaving behind basic alkaline blood. Then the other thing is ingestion of alkaline substances like a bunch of Tums or Drano, whatever. To our final slide. And this is actually something that we're going to keep up. I think with this slide and then the diagrams that we have on the board, we should be able to walk through this case study relatively easily. I say relatively easily. Let's give it a try. We're going to do the one that it has the acid-base problems first, and I'm going to walk us through the first one. I'll probably need a little room. I don't know where I'll have room to write this. I likely will have to erase this stuff. Is it everybody okay if I erase that picture? I'll get that out of the way, and then I'll go through the first. Maybe we can shuffle it and just pick a random person out of this case study to start with. And then after we do this, we'll run through the diarrhea case study. Does anybody need a copy of this real quick? Anybody else? All right. Let me get some space cleared. So I'm just going to leave this on the board so we can all just read through the case study. I'll keep it simple and I'll just do the first one on the very beginning page, and then we can break up into some teams to see if we can get through the rest of them. Each one of these scenarios, there's 4 people in this case study. We're always going to start with these same 3 steps. So don't get too far ahead of yourself, otherwise it will be confusing. Here's how I'll demonstrate how to do the first one. The first thing that we're going to look at, and I said this just a second ago, is you simply just look at the pH of this individual's blood. If it's below this normal range, we're going to immediately know whether their blood is too acidic, or if it's above the normal range, their blood is too alkaline. That tells us what acid-base problem they have right off the bat. They're either in acidosis or alkalosis. What we don't know, however, is why their blood is too alkaline or too basic. Is it due to metabolic reasons, or is it due to respiratory reasons? And to answer that last question, we need to move into step 2 and step 3. But we don't ever want to just jump to step 2 or step 3. Here's what I mean. So let's go through this one at a time. I'm going to start with the first one, which is Sean. And help me out here. It says Sean has a blood pH. Let's just write this down so that we know we're talking about Sean. Sean has a blood pH of 7.60. It tells us his partial pressure of carbon dioxide is also 20 millimeters of mercury. And what else do we have? We can see that his blood levels of bicarbonate is 19. It tells us in here whether Actually, it tells us right here whether that is above normal or below normal. You don't have to memorize these things. This is something that will be given to you. So this is something that we will refer to as we work through these problems. So without fearing that you have to remember these numbers, just reference them. Let's look at Sean's conditions and let's try and answer some of these questions. So the first question is, what acid-base imbalance does Sean suffer from? And the first step that we're going to do is we're going to look at the pH. So is he in a situation where his blood is too acidic or too alkaline? He's in alkalosis. But we don't know why. We don't know why his blood is too alkaline. So we need to answer this part. Why is it too alkaline? And for that, we need to go into step 2. So, the second question is like, what are the possible problems? What are the cases? What are the possible causes of his blood being too alkaline? Here's where we get into what's going on. So if we look at the partial pressure of carbon dioxide, normal ranges are anywhere from 35 to 45 millimeters per mercury, and it looks like he is at 20 millimeters of mercury. So this is below the normal range. And if we go over here, this tells us if the partial pressure of carbon dioxide is, here it is, if it's below normal, respiratory alkalosis is what's going on. And his partial pressure of carbon dioxide is certainly below 35. His blood is too alkaline. So we've solved this. Now we can go back and we can say his blood is too alkaline due to respiratory reasons. So respiratory alkalosis is what's going on there. We could now answer this second question. What are the possible causes of the acid-base imbalance? First one says abnormal CO2 retention. That's hypoventilation. That's talking about the lungs making the blood too acidic if you're holding on to CO2. Letter B is excess loss of CO2 from hyperventilation. His blood is too alkaline, and it is the lungs that are causing this. So B looks like a likely option. Severe diarrhea, well, that would make the blood too acidic, but for metabolic reasons. And in fact, if we see, we can stop at number 2 because we've identified that his blood is too alkaline for respiratory reasons. If we were to go to number 3, for example, this is telling us whether the blood is too acidic or alkaline for metabolic reasons. His blood level of bicarbonate is 19. Normal levels are 22 to 26. He's below that. So if you're below those levels, that's likely due to metabolic acidosis. He doesn't have acidic blood. He has alkaline blood. So this scenario down here doesn't fit, which would make sense. This one up here did fit. Respiratory alkalosis. We figured that out. And what we can make sense out of this, if his blood is too alkaline, then it would make sense that he's also trying to get rid of the amount of bicarbonate. There's already not enough free hydrogen ions in the bloodstream. So like we were seeing over here, if you have too much of this bicarbonate in the bloodstream, we don't want to hold on to more bicarbonate. We want to get rid of that bicarbonate. Okay, so we've answered at least the first 2 questions for Sean. If we get into number 3, there's the 3-step scenario that we mentioned. Chemical buffer systems. If his blood is too alkaline, would those chemical buffer systems, that bicarbonate or carbonic acid over here, would that tend to give up? You can either, here's also what's sometimes confusing, that box where it's talking about chemical buffers. Let's pull this up so you know what I'm talking about. This first box right here where it's talking about chemical buffers, this term give up, you can also write donate next to the word give up. Let's make that a little bigger. So give up or donate, that means the same thing. Take up or accept, that also means the same thing. So if we're in a situation where the blood is too alkaline, how are those chemical buffers going to handle the free hydrogen ion? Are they going to donate / give up, or are they going to accept, take up one of those hydrogen ions?
Bloodstream?
Let's draw this. Let's see where we're at. So we're thinking of these, that carbonic acid that we talked about in the bloodstream. So let's draw our capillary. In this case, we've got a situation where there is way more bicarbonate than there are these hydrogen ions. This is kind of drawing what Sean is dealing with. He's dealing with this alkaline blood. And I guess our question here, if we're thinking about these chemical buffers, this carbonic acid, is this thing in a situation where the blood is too alkaline, I guess we're asking, is this thing going to end up getting rid of a hydrogen ion, or is it going to end up picking up a hydrogen ion? Remember, this carbonic acid goes back and forth between holding on to an extra, here's its form where it has an extra hydrogen ion, and the other form over here is after it's kicked off that hydrogen ion. So it's constantly either breaking into two pieces, or these two pieces are coming together to form just one molecule. So this whole thing here is our buffer system. And that buffer system can go back and forth. Either, if it goes this way, then it's basically picking up a hydrogen ion from solution. So if we got one out here, and this hydrogen ion mixes with this bicarbonate, they go to carbonic acid. But if we're going the opposite way, and this carbonic acid wants to lose one of these hydrogen ions, as it gets rid of this hydrogen ion, it leaves behind just the bicarbonate. So this buffer system, it can go back and forth, either getting rid of one of these hydrogen ions, like we can see it doing now, or picking one up. And I guess I'm kind of giving it away here for Sean. He doesn't have enough of these free hydrogen ions in the bloodstream. So the chemical buffers are going to be doing everything they can to add these hydrogen, give up these hydrogen ions, or donate these hydrogen ions to the blood plasma. The next thing on the next box here asks us, how would the respiratory system compensate in Sean's case? Trick question alert. What are we thinking there? How is Sean's respiratory system going to compensate? We're talking about Sean's respiratory system, maybe not everybody's respiratory system. It wouldn't be B, not for Sean. Why wouldn't it be B for Sean?
Is it because he's already hyperventilating?
Well, like we just said, if the lungs are the problem, the lungs can't compensate for a lung problem. So that's where I said trick question alert. Sean has respiratory alkalosis. So his second line defense there, the respiratory system, isn't going to be able to help for a respiratory problem. So that's where the answer for Sean here for the respiratory system would be C. The respiratory system can't compensate for this problem because it's causing the problem. So you're right. So then that takes us to the renal system, which can respond. We have type A and type B intercalated cells. One is going to conserve either hydrogen ions or bicarbonate. Which one of these cells would respond for Sean? Type B intercalated cells, because his blood is too basic. And that would cause, so those type B intercalated cells, what would they conserve? Would they conserve, and by conserve we mean prevent ending up in the urine. This stays in the bloodstream, Secrete means end up in the urine. We're getting rid of that. We're getting it out of the body. Type B intercalated cells, which would they want to hold on to? Type B cells would want to hold on to that hydrogen ion because the blood is already too alkaline, and they would want to secrete that bicarbonate because there's already too much bicarbonate in the bloodstream. We got that. And then the last thing down there, it's asking about Sean's urine. Would his urine be acidic or alkaline?
After all this went through?
That's right. You're talking about right now, right? Even in the middle, like we said, if the blood is too alkaline, the urine is too alkaline. The urine matches whatever the blood is. If the blood is acidic, the urine is going to be acidic. Yeah.
I'm having trouble understanding.
Okay. What are you thinking?
It says here if his blood bicarb is 19, and that's low.
That's right.
Why is it acidic? Like, more acidic?
Bicarbonate levels are low. This is the body's way of trying to get rid.
Or sorry, why is it basic? Sorry.
What's
If that is low, why is like the blood more basic? Alkaline.
so that's it. So pH isn't necessarily have to do with bicarbonate levels. pH is the pH of the blood has to do with just these hydrogen ion concentration levels.
Because I know that the blood CO2 levels will make it, alkaline, but why the blood? Like, why is the blood alkaline as well? I don't understand that.
So this blood is alkaline because it has too few of those hydrogen ions in here. I think me drawing a bunch of these bicarbonate ions in here is maybe getting you to focus on these bicarbonate ions. And really, what I could have drawn chloride ions in there, any kind of negatively charged ion. We could have put other positively charged ions in there as well. pH is really just concerned with how many of those free hydrogen ions we have. And if there's a low amount of those hydrogen ions, we consider this alkaline blood. And if there's too many of those free hydrogen ions, it would be very acidic.
Because on here, it says that if it's less than 22, it's metabolic acidosis.
That's right. So let me pull back these things up real quick. Because what you're doing is you're jumping to step 3. And you're jumping right over step 1. Right? And I said you're going to get in trouble if you jump down here to step 3 and you go, oh, his bicarbonate is below this. Why does this not work? And this would only be applicable if we're dealing. This would explain why somebody's blood would be too acidic. And again, this isn't meant to explain respiratory acidosis or alkalosis when we go in a backwards scenario. So that's to avoid asking those or thinking in that type of way. We want to always start at the first step and then work our way down. That's kind of how this slide is built. So starting from the first thing and knowing that Sean's blood is above, it's in the alkalosis range, we're going to ignore anything else on this slide that says anything with acidosis. And the reason, like if we want to make sense as to why does his blood, and I just removed the case study, this tells us his blood bicarbonate levels are low. This is going to be one of the ways that the body is trying to help compensate by getting rid, I guess, of the extra bicarbonate levels. When we're thinking of the pH of the blood, though, we really want to focus on the individual hydrogen ions. Let's work through a couple to see how when we get to metabolic acidosis and metabolic alkalosis, seeing those scenarios might be more helpful than just making it through the first one. So what we saw in this situation was that his blood was too alkaline. There wasn't enough of those free hydrogen ions, so the urine is also going to be alkaline. Let me get another color, and let's try the second one, and I'll let you walk me through this just by me asking the questions. And maybe we can split screen this. Let's try and pull this one. I'm going to go to the second one, which is Grace. We can see Grace's numbers over here, and maybe I can do it like this. Well, it's not going to let me make it any bigger than that, I don't think. There we go. Okay, so let's do it like this. Okay, so now we can read the whole thing. I'll grab my pointer and we'll just grab a different color. We're looking at Grace now, so I'll put Grace in this color. Grace has a situation where, and that's why it's helpful to write this down, the pH of Grace's blood is 7.2. Let's separate her from everything else that's going on. So right off the bat, we can say that what type of imbalance is she suffering from? Some type of acidosis. But we don't know why. We don't know the first part to that acidosis. And we need to figure that out before we can see why the blood is too acidic, before we can answer question number 2. So to see why the blood could be too acidic, it could either be too acidic due to respiratory reasons or it's too acidic due to metabolic reasons. So now that we've gone through the first step, let's see which one of these next 2 steps do we even need to pay attention to. Because if we pay attention to the wrong one, it's just going to confuse us. So we know we're looking for one of these 2 scenarios that has to do with the blood being too acidic. So we either have respiratory acidosis or we have metabolic acidosis. It's going to be one of these 2. So we're looking for the numbers that fit. Let's go to the next number for Grace. The next number for Grace is that she's got a partial pressure of carbon dioxide of 31 millimeters of mercury. That falls below the normal range, and below the normal range is respiratory alkalosis. Does this fit with Grace's situation? No, so this doesn't work. We got to move down to step 3, and we're looking for something that explains why her blood is too acidic. And the next number we have to go off of for Grace is bicarbonate levels, and bicarbonate levels are 13. That is well below normal. So this one fits for her, and it also explains why her blood is too acidic. So now that we've found the one that matches, this is the reason her blood is too acidic. It's metabolic acidosis. Right, so if we go back to 31 millimeters of mercury, that doesn't fit here at all, right? This saying if it's above, so that's kind of that scenario. If you tried to explain it with this, it doesn't make any sense.
That one, the blood is not what's causing her pH levels to be like that.
That's right. That CO2 tells us that it's the respiratory that was the problem. So now we figured out why the blood is too acidic. It's metabolic reasons. So metabolic acidosis. And now we know, now that we've answered that it's metabolic acidosis, which one of these things explains metabolic acidosis? C, either severe diarrhea or being diabetic. Those are metabolic reasons the blood can be too acidic. So in that case, going back to our chemical buffers, would those chemical, if the blood has too many of those hydrogen ions, would those chemical buffers try and give up more hydrogen ions or take up some of the excess hydrogen ions? Maybe I'll change the, because I think it's the way those words are there. Chemical buffers, if there's already, in the case of Grace has way too many of these hydrogen ions in there already. So these chemical buffers are, notice how the chemical buffers aren't saying whether they're giving up or donating this bicarbonate. It's just about those hydrogen ions. So if she has too many of those hydrogen ions, these chemical buffers are going to go this way. They're going to try and pick up this excess hydrogen ion, bottle it up in the carbonic acid molecule so it's not just floating around there free. If we had another one of these buffer systems over here, maybe it picks up this hydrogen ion. So these buffer systems, if the blood is too acidic, they're going to be taking up, accepting these hydrogen ions. Because we want to try and reduce how many of them are just freely floating around in the bloodstream. Let's go to the next one. The respiratory system. No trick question alert here. How would the respiratory system help Grace out? She's got too many of these hydrogen ions in her bloodstream. So in that case, we said that, if I can get this off, water and CO2, they lead temporarily to this carbonic acid that breaks apart into that hydrogen ion and that bicarbonate. So there's a direct relation. The more CO2 we have, the more of these free hydrogen ions we could end up with. So in Grace's case, she's got too many of those free hydrogen ions, which means that she's already got plenty of this carbon dioxide in her bloodstream. One way we can reduce the number of free hydrogen ions, because let me catch that for just a second. If her blood was too acidic for respiratory reasons, then she would have too many of these free hydrogen ions because she has gotten too much carbon dioxide in her bloodstream. In this case, her blood is too acidic due to metabolic reasons. She's diabetic or whatever it might be. So she's got a bunch of she's got a bunch of hydrogen ions in her bloodstream. The lungs can compensate for that. So one way that the lungs can decrease hydrogen ions is by adjusting CO2 levels, by decreasing those as well. So how would the lungs decrease the amount of CO2 in the bloodstream?
Pulmonary ventilation.
That's right, increasing that pulmonary ventilation, hyperventilating that. So pulmonary ventilation, as that increases, you're blowing off the CO2. And as you're getting rid of that CO2, you have fewer free hydrogen ions. So now we're at the renal system. Which one of those cells, those intercalated cells, would respond for Grace? Yeah, so her blood is too acidic, so type A cells are going to respond. And those type A cells would conserve what? I think I heard it. Type A cells, there it is over there. Type A cells, they're getting rid of something and they're conserving something else.
Oh, so the conserving is like what's going back into the blood?
Yeah. Here are those type A intercalated cells. They're getting rid of the hydrogen ion. We want to urinate that out of the body because we already have too many of those if the blood is too acidic. And we want to conserve this bicarbonate because that bicarbonate, like we were just showing over here, this bicarbonate could end up combining with this hydrogen ion and locking it away into this carbonic acid molecule. So that's how those type B and type A intercalated cells can adjust the pH. In this case, their blood is too acidic, so it's type A cells secreting hydrogen ions and conserving bicarbonate. And what would we expect Grace's urine to be? Would it be acidic or would it be alkaline? That's right, her blood is too acidic, and if we look at what's going on, those type A intercalated cells are getting rid of hydrogen ions in the urine, so that urine is also going to be acidic. Feel better about it after the second one? Let's try a third one to see if we're feeling even better now. This will confirm it. So George, what's going on with George? Let's put him in a green color. George's pH of his blood says it is 7.54, if I'm reading correctly. So we know right off the bat, so that we don't confuse ourselves, we go to the first step. And we can see that he is above the normal range. So for George, we know that his blood is alkaline. But we don't know why. We need to investigate further. So let's see why his blood could be alkaline. And so that narrows us down to either step 2 or step 3. We're only concerned with the options that tell us alkalosis because acidosis does not apply here. So one of these is going to work and the other one isn't going to apply at all. We'll first go to step 2 to see if that works, and if not, we'll move on to step 3. And the second number up here for George is his partial pressure of carbon dioxide is 52 millimeters of mercury. That's above the normal range. Above normal ranges of millimeters of mercury for carbon dioxide, that tells us respiratory acidosis. Does that explain what's going on with George? No, it doesn't fit. His blood is not acidic, his blood is alkaline. So let's keep going to see if the last scenario fits. And for George, it tells us his blood bicarbonate levels are 36, which is well above normal. Let's see what happens in above normal cases of bicarbonate. Well, one scenario there would be metabolic alkalosis. And sure enough, that explains his blood is alkaline, and we've now figured out it's due to metabolic reasons. So let me, before I forget, label metabolic alkalosis. Which one of those scenarios we said can lead to metabolic alkalosis? It's one of the options here. The blood is too alkaline for metabolic reasons. Sorry, what was that? Oh, it's coming from over here. Sorry.
D.
D. Yes, that's why I was expecting the vomiting or something like that. But you're right. Letter D is what's going on there. So prolonged vomiting or eating like a whole roll of Tums or something like that, ingesting those alkaline substances would be a metabolic reasons your blood is too alkaline. So D for this one, and then when we get into the chemical buffers, if the blood is too alkaline, it doesn't have enough of these free hydrogen ions in it, very low levels of hydrogen ions, would those chemical buffers donate more hydrogen ions, or would they wrangle up and pick up any of the ones that we have? They would want to donate more. So these chemical buffers, there it is. Get my chemical buffer picture out of the way. So this chemical buffer goes back and forth. It can either donate or accept a hydrogen ion. And if we're in a scenario where there's not enough of these free hydrogen ions, it's going to be kicking out and donating excess or extra hydrogen ions to compensate for the fact that there's not enough of them in there. What about the respiratory system? Is there a trick question alert here? I don't know. What would the respiratory system do for George?
Does it matter if would it increase?
I would say D.
Decrease pulmonary ventilation.
Well, it is going to do something because the lungs are not the problem here. It's the metabolism that's the problem. So the lungs will be able to compensate and do something. And if there is not enough hydrogen ions in the bloodstream, this is what I was just kind of doing with this chemical equation that shows there's a direct relationship between how much hydrogen ions we have in the blood, how many free hydrogen ions we have, and the levels of carbon dioxide. And so for the case of George, there's not enough of those hydrogen ions. There's very low levels of hydrogen ion for him, which means there's very low levels of carbon dioxide. One of the things that we could do to increase hydrogen ion levels is to increase carbon dioxide levels, and that's going to give us some more of these free hydrogen ions. Just like the chemical buffers are trying to give us more hydrogen ions, the lungs can do this by holding on to CO2. As long as we can increase CO2 levels, we should be able to increase these free hydrogen ions. So now back to this one. Which one of these options is going to increase carbon dioxide levels? B, yeah, okay. It's hard for me to hear things, I guess. Pulmonary ventilation decreases. So this would be, you're not exhaling as frequently. So that carbon dioxide is just building up in your bloodstream. B. Now the renal system, let's scroll down a little bit. George is in a situation where his blood is too alkaline. So which one of the cells of the kidney are going to kick in? And here we have a picture of those kidney cells so we can think about it. Type B are going to help George because George's blood is too alkaline. And it says those type B cells, what are they going to conserve? Here's we don't want to get rid of that hydrogen ion. We want to conserve that and bring it right back into the bloodstream to neutralize all those bases. And then what we secrete is the base. And since we're secreting base into the urine, George's blood is too alkaline, too basic, and we can see that his urine is now also going to be alkaline and basic because that's how the kidneys are compensating for the fact that his blood is too alkaline by getting rid of those bases. And so that's why the urine becomes also basic. I don't know why I'm grabbing this anymore. Let's go back to the last one. So now after the third one, hopefully we're feeling a little better. Any questions about that one? Yep.
So that 52 millimeters of mercury would increase because holding on to, like holding your breath or whatever? Is that what's happening?
That's exactly how you're saying. So when we're seeing that in the case of George, his blood is too alkaline for metabolic reasons. And one of the things that you can do, since the lungs are not the problem, the lungs can hold on to more CO2 in an attempt to increase hydrogen ion concentration. And the fact that the carbon dioxide partial pressure levels are so high in George, you're seeing that his lungs are attempting to compensate for that. That's what that is. And like we were saying before, it didn't make sense because the lungs couldn't compensate if it's a metabolic issue. So that was not making sense there. Let's see the last one. Sharon. Let's put Sharon in I bet she would like a red color. I could be wrong, but let's try that. Sharon, let's see, she has a situation. We've got just enough room to write Sharon's information down here. Her pH of her blood is at 7.24. So we know right off the bat Sharon is dealing with a situation where her blood is too acidic. So acidosis. But we don't know why. Why is this blood too acidic? Let's dive in a little further to see that Sharon has a partial pressure of carbon dioxide of 60. That's up there. Her blood bicarbonate levels are at 39.5. So that we don't confuse ourselves, we can't forget her blood is too acidic. So we're looking for one of the either step 2 or step 3 that explains acidic blood. So let's go partial pressure of carbon dioxide above normal ranges. So above normal ranges, which this certainly is, says respiratory acidosis. And sure enough, her blood is too acidic, so we don't even need to go to step 3. And if we did go to step 3, it wouldn't make sense. Try it if you want. Won't make sense. So we can stop after step 2, and we've identified that it's the respiratory system that's causing the problem here. Respiratory acidosis. If the lungs are the problem here, then trick question alert, these lungs are not going to be able to regulate CO2 levels to help compensate for hydrogen ion levels. So if we've identified that it's the respiratory system that's causing the blood to be too acidic, what would be one of those possibilities? Respiratory acidosis. That's right. I guess I don't know what I was saying here, but that is correct. So in the case, her blood is too acidic. So that would mean too many of these free hydrogen ions tells us there's lots of carbon dioxide in the bloodstream. So sure enough, if we look at partial pressures of carbon dioxide, they're through the roof. Those high levels of carbon dioxide are causing our high levels of hydrogen ion concentration. So abnormal retention of CO2 is what's causing this. So we can move to the box. Those chemical buffers, if there's already too many of these hydrogen ions here, what would those chemical buffers do? They would try and pick up those hydrogen ions. Take them up. What about the respiratory system? How is it going to help Sharon? Can't do nothing. It's the problem. It's the reason why there's too much hydrogen in the bloodstream, because it's retaining CO2. She's not able to blow off CO2. Maybe she's got emphysema. That would be an example. So the lungs can't compensate. She can't really adjust pulmonary ventilation. So all her eggs are put in the renal compensation basket. And hopefully type A and type B cells are around. If we're thinking about Sharon's situation, we know those chemical buffers are going to be taking up hydrogen ions. The respiratory system can't compensate, but how would the kidneys compensate? Which one of those cells in the kidney? Did I hear type A? Yeah, type A intercalated cells. And we can see those type A intercalated cells are going to conserve, what do they conserve? Bicarbonate, and they get rid of those hydrogen ions. That would make sense. She's already got too many hydrogen ions in the bloodstream. So the type A intercalated cell is getting rid of those hydrogen ions. So type A, conservation of bicarbonate, secretion of hydrogen ions, and then we would expect Sharon's urine to be either acidic or alkaline. What would that be? Acidic. Her blood is acidic. Type A intercalated cells are activated because the blood is acidic, and we can see hydrogen ions being pumped into the urine, so the urine is going to be acidic.
How would you test, well, for the pH level, for your blood pH level, how would they test that? Just like urine sample? Or do they take your blood and then just do like a test strip? See if it is acidic?
I would think test strip would be one of the main ways of doing that, but there might be other ways. You could certainly indirectly check through the urine.
Yeah.
That would be how we would probably do it in a class, so that we wouldn't draw blood.
If Sharon was a normal person and that didn't have like all these problems or whatever, would she just breathe in more, like more pulmonary ventilation to fix that problem?
Sharon's problem is that her lungs, she's got like emphysema in this case. So she's not able to exhale normally. And so there's just going to be a slow buildup of carbon dioxide in the bloodstream.
For a normal person, it would just increase the rate of breathing.
That's right. The lungs could compensate. So, that's one of the reasons.
I was getting stuck. That distinction is where I was getting stuck.
But that's why, once you connect the dots between the renal system and the respiratory system, controlling the pH of the blood, you can see why it's so problematic for somebody to deal with COPD or emphysema or some of these respiratory issues, because that's really your second line of defense. If the chemical buffers get overwhelmed, that has to be there to adjust carbon dioxide levels and pH levels of the blood. It's in that very narrow range that is normal. Otherwise, you'll see inadvertently, somebody's respiratory system start to hyperventilate. And I used to freak out when I would see my dad do that, but understanding doesn't make me feel a whole lot better about it, but at least you understand kind of what's going on. Any other questions about that stuff?
So somebody with emphysema or COPD, since they don't have the secondary defense, the respiratory system, it would just be up to, obviously, the chemical buffers. But the renal system, would that cause premature renal failure or something?
It can certainly be harder on the kidneys. What you really lose is that in that second line of defense, you lose the compensatory mechanism that can work on the order of minutes, like 30 minutes or 40 minutes. So if my dad, or we'll say my dad has the respiratory system, so luckily when that kicks in, it brings him back into that normal range. But if somebody had COPD or something like that, and their blood got out of whack for whatever reason, their only compensation would be if their chemical buffers got overwhelmed, their only compensatory mechanism would be the urinary system, and that could take hours to days, which that might be too long.
Yeah.
So you're at there's a lot more care that has to be taken, I guess, because you have to worry about those other things. You don't have that lung compensatory mechanism.
So if somebody was diabetic and had emphysema, they'd be in
Double whammy.
Yeah.
Comorbidities. Right. Hope nothing else messes up that delicate balance there. That's why people that are struggling with those things, they get hit with something like COVID, and it's even, the body is already doing so much.
Yeah.
So in alkalosis situations, your chemical buffer will give up the hydrogen.
That's
and
right.
For acidosis, your chemical buffer will take up the hydrogen.
Correct. And we only have a finite amount of those chemical buffers, so their adjustment is immediate, but it's minor. It's quick, but there's not a lot of it. So once we run out of those chemical buffers, then it's the lungs that are the next to kick in if you got them. And if you don't have them, then you're at a severe disadvantage, because we're now waiting on those intercalated cells to kick in. If you're ready for some more fun, we've got, and I realize I've used up my...
Sorry, we were asking questions.
No, you're great. This is what it's all about. So hopefully that was helpful. The second thing that we see is also, does anybody need the diarrhea case study? You already kind of know where we're going with this. This is somebody with diarrhea, and you might start to guess at what type of acid-base imbalance that causes. And I'll pull this one up just so that we can read it. Let's blow this one up a little bit. Okay, here's our diarrhea case study. This is involving a lady named Melanie who wedded the man of her dreams. It was a wedding that took place, there was a honeymoon in Mexico. Melanie, has severe traveler's diarrhea because she drank the water down there. Despite attempts to control the diarrhea with over-the-counter medications, she continued to have about 8 to 10 watery stools daily, so that tells you she's losing water. She's losing those bases in the stool as well. That's not being reclaimed. She progressively becomes weaker. On the third day, she's taken to a local emergency department. On physical examination, her eyes were sunken, her mucous membranes were dry, her jugular veins were flat, tells you that she's severely dehydrated. She was pale, her skin was cool and clammy. The physician ordered laboratory tests, and you've got the results below. This is just a case study. You don't have to remember all of these things, but it's good practice. It tells us that the first laboratory results that we can see show us that her blood pressure, when she was sitting up even, or even when she's supine, kind of laying down looking up at the ceiling, it was 90/60. It should be somewhere around 120/80. So her blood pressure is severely low. What might be causing her blood pressure to be too low? That's exactly right. Blood volume and blood pressure are directly related. So the fact that she's got severe diarrhea and has been losing blood volume tells us why her blood pressure is so low. Okay, so what about why we can see that her blood pressure is really low, but her heart rate is well above the normal 76 beats per minute. It's about 120 beats per minute. This causes us to think back to stuff that we mentioned in previous units. Actually, we mentioned it in this unit too. We said when blood pressure, right, when blood volume and blood pressure drop too low, something happens. The body responds by producing something that boosts blood pressure and blood volume. What was that? There was, I won't pull up the slide, but
Is it renin?
yes, it's renin. We said when blood volume drops too low, that that causes blood pressure to drop too low. And when blood pressure drops too low, there are those cells that we call the macula densa cells. We said that blood volume and blood pressure were also directly related to filtration rates. And when filtration rates dropped too low, we actually saw it last time in that juxtaglomerular apparatus. It's the group of cells next to the glomerulus. Those macula densa cells, they were able to detect if filtration rates dropped too low, and they boosted filtration rates by boosting things like blood volume and blood pressure. One of the things that they did to boost blood volume and blood pressure, it was renin that was produced first. Renin led to angiotensin 1, that led to angiotensin 2, and angiotensin 2 did multiple things that all boosted blood pressure and blood volume. One of those things that angiotensin 2 did was stimulate sympathetic nervous system. That led to the blood vessels constricting body-wide. That boosted blood pressure because the volume decreased. But in addition to the sympathetic nervous system causing the blood vessels to constrict, it also increases heart rate. So this elevated heart rate tells us that sympathetic nervous system is activated. It's trying to do whatever it can do to boost blood volume. We can also see respiratory rate here, which would normally be around 16 breaths per minute, is at 24 breaths per minute. And these are deep breaths. So this is, she's doing something like that. What's going on there? Well, maybe without me, all these questions are below. But the first question is, what acid-base disorder does Melanie suffer from currently? What would that first answer be? Yeah, I guess it's about to get there. It tells us that her pH of her blood is 7.25, so we know that the blood is too acidic. And it told us up at the top that she had severe diarrhea for multiple days. So she has lost all of the bases in her stool. She's lost water and she's lost bases, and it's left behind more acidic blood. So we could say that Melanie is one of these situations where she's suffering from metabolic acidosis. The blood is too acidic for metabolic reasons. In this case, prolonged diarrhea. One other thing that's notable that's a little bit weird, well, it's not weird, it's just something that we should point out, is that when thinking about what Melanie is dealing with, we just said that her sympathetic nervous system is stimulated, and we can tell that because her heart rate is so high. That's also one of the ways the sympathetic nervous system tries to compensate for low blood pressure. If the heart rate increases, that increases cardiac output. That might boost filtration rates. The side note over here, remember these principal cells, these are the ones that are responsible for absorbing extra sodium, absorbing extra water. This helps boost blood volume and blood pressure, so that renin, angiotensin, aldosterone system certainly used, well, like its name suggests, aldosterone to boost blood pressure and blood volume. I said there was something that could be problematic with aldosterone. If you have prolonged activation of aldosterone, you are conserving sodium ions. That helps boost the blood pressure and blood volume, but you're also getting rid of excess potassium ions. If we look at the last thing here, it shows that the blood levels of potassium for Melanie here are actually below normal. That might not normally make sense. In fact, if the sympathetic nervous system was not activated, this wouldn't be the case. Her blood is too acidic. So when the blood is too acidic, what happens is we're typically getting rid of these hydrogen ions and holding on to extra potassium. In the case of aldosterone, aldosterone is going to do the opposite. It's going to start getting rid of potassium ions. So the thing down here that you might not think fits this low potassium levels, it actually does fit if you remember that sympathetic nervous system is stimulated, and with sympathetic nervous system stimulation comes this aldosterone, with aldosterone working, it gets rid of a lot of potassium ions. So that's what leads to this electrolyte imbalance, where you got a lot of sodium, but very little potassium. So you can see that she was taken into the hospital. She was treated with strong anti-diarrhea medication so that she stopped losing water and losing those bases. She was given an IV infusion of salt water. This sodium chloride, this is meant to mimic the salinity of normal cytoplasm, normal body fluid. So this infusion of salt water IV is really just meant to boost her blood volume. By boosting blood volume, that is going to help boost blood pressure and filtration rates. The second part, this potassium bicarbonate, that potassium bicarbonate is going to break apart. If we just look at potassium bicarbonate, this thing is going to break apart into a potassium ion, and then the other thing is a bicarbonate. And for the case of Melanie, she's in a situation where she's got metabolic acidosis. There's too many of those free hydrogen ions, so this bicarbonate is going to help combine with some of those free hydrogen ions. It locks that free hydrogen ion up into this carbonic acid molecule. So one of the reasons she's given this potassium bicarbonate is to neutralize those free hydrogen ions. And the other side note with that, she's already got too low potassium levels due to aldosterone working. So this potassium bicarbonate helps replenish potassium that she's already low in, and it also helps neutralize the excess hydrogen ions that are in her bloodstream. She's got acidic blood. So I think we went through all of those things. Let's see. So I think this is redundant because we did answer those, but help remind me, what is it that she suffers from, acid-base disorder? Metabolic acidosis. Her blood is too acidic, and it's for metabolic reasons. In particular, the diarrhea caused it. But in the second question, how did that diarrhea cause that acid-base disorder? I said it a couple times, and it reminds us of what the large intestine does.
It was losing the basis in her stool.
Yeah, if the large intestine doesn't work properly, then you lose the water and you lose the bicarbonate base that was secreted. I really have used up almost all of my space here, but the pancreas is going to release this molecule. It's sodium. I think we even saw this last time. It's sodium bicarbonate. That's what the pancreas releases. Some of that breaks apart in just a sodium ion, which our body can deal with, and the other part is that bicarbonate that we've seen a bunch of. And this is what neutralizes that stomach acid. It ends up in the large intestines and would be lost in the stool if the large intestine didn't reclaim this with that excess water as the stool gets compacted. So the diarrhea caused this because what she lost in that diarrhea was the bicarbonate base. 3, it says, why do you think there was an increase in the depth and frequency of Melanie's breathing? How would we explain that hyperventilation? 24 breaths per minute, and they were deep breaths.
To get rid of the CO2? Or
She's trying to blow off the CO2. That's the lungs' way of trying to compensate that the blood is too acidic, so it's trying to blow off that CO2. And we answered this one, I believe, is why was Melanie's blood pressure lower than normal? That was the first thing. I think Ebony answered that one. Or it came from back there somewhere, maybe. Amanda, did you answer that? Why was her blood pressure lower than normal? I've already covered it up in here, but remember, blood pressure is tied to blood volume. Dehydrated. That severe dehydration, she's lost her bases and she's also lost the water in the stool. So loss of blood volume leads to that loss of blood pressure. And that loss of blood pressure causes the kidneys to release renin. It stimulates that renin-angiotensin-aldosterone system. And one of the side effects of that renin-angiotensin-aldosterone system, we said the sympathetic nervous system gets stimulated. So the answer to number 5, why is her pulse rate elevated? It's because that renin-angiotensin-aldosterone system has stimulated the sympathetic nervous system. Increased heart rate, constriction of blood vessels, all things that try and boost that blood pressure and filtration rates. I think I just answered number 6. How would you expect Melanie's renin-angiotensin-aldosterone system to be affected? Well, it would be stimulated. It would be ramped up. Its activity would be increased. And like we said, it does, that renin-angiotensin-aldosterone system does multiple things, one of which is gets that sympathetic nervous system activated. Okay, so that explains why the pulse rate is elevated. Why would her potassium concentration levels be low? I think I answered that one too. But it kind of ties into number 6. You just said that the renin-angiotensin-aldosterone system would be cranked into high gear. One of the A's of that renin-angiotensin-aldosterone system is aldosterone. And if aldosterone is working, it's this revolving door that holds on to sodium because it's trying to boost blood volume, but at the cost of potassium. Low levels of potassium tell us that that aldosterone system has been working for too long. It would end up leading to this imbalance, electrolyte imbalance, where you got too much sodium and not enough potassium. Those neurons might not be able to repolarize. They have to be able to shun lots of potassium very quickly to repolarize. And if they don't have enough potassium to get rid of, cells can't repolarize. This could be fatal if neurons can't communicate. Which is one of the, kind of leading into the last one here. I think that's the last one. What was the rationale for treating Melanie with an infusion of sodium chloride and then potassium bicarbonate? I gave that away. The sodium chloride, this mimics normal salinity of blood fluid. So you could give an IV that has a saline solution just to boost blood volume without making that blood more concentrated with solutes or watering it down. The saline IV is just there to adjust blood volume, which is going to help adjust blood pressure and filtration rates. The other part of that, the potassium bicarbonate, the rationale for that was that we know that potassium levels are too low. So by giving some potassium bicarbonate, that's going to replenish the low potassium levels. And then the other half of that was the bicarbonate. That's going to be helpful because it's going to immediately start to combine with some of those free hydrogen ions and further reduce the acidity of her blood, preventing those, as many of those free hydrogen ions from floating around. Thinking ahead, this is really all of our urinary physiology discussion. Next time we have just a review, then we'll have our exam about urinary and digestive, and then when we get into the 5th unit, it's just reproductive system. There's not a whole lot of chemistry on the reproductive system. I went through, I think, all of the models for digestive and respiratory last time, so if you missed any of that stuff, I'll be glad to repeat the models. But that completes our lecture for urinary stuff.